Hello, and welcome to my poster presentation on Kisspeptin's role in bone cell differentiation and growth. My name is Jordan Matz, and I want to thank you for listening. First, I want to give you some background information about Kisspeptin, and then I will discuss the objective of my study, go into some preliminary data, and end with talking about the future plans I have for this project. Kisspeptin is a protein that communicates with cells via a G-coupled protein receptor called GPR54. Kisspeptin is most notable for its role as the principal regulator for the onset of mammalian puberty. And when kisspeptin or its receptor are disrupted, it leads to the disease state of hypogonadotropic hypogonadism. Kisspeptin neurons are localized to very specific regions in the brain, but they signal to a wide variety of regions in the body. While most previous studies focus on the relationship between kisspeptin, puberty, and reproductive physiology and pathologies, very recent studies have investigated the potential role that kisspeptin plays in bone. In fact, a recent study from 2018 found that the kisspeptin GPR54 signaling pathway induced differentiation of a specific osteoblast-like cell line, which led to the conclusion that kisspeptin may actually act directly on bone to induce osteogenesis. This leads us to our present study. So using a different osteoblast-like precursor cell line than the previous study, I wanted to know if something similar could be replicated and hypothesized that kisspeptin and or its receptor are expressed by bone cells and provide a communication pathway to regulate bone growth and differentiation. This study is still very much so a work in progress. However, I do have a bit of preliminary data. I cultured the osteoblast-like precursor cell line MC3T3E1. I harvested those cells, extracted the total protein content, and performed Western blot analysis. I probed with an anti-kisspeptin receptor or GPR54 antibody. As the figure shows, I was able to see expression of GPR54 at baseline conditions in this cell line with both a loading dose of 15 and 30 micrograms. This was pretty exciting to me since GPR54 has never been shown to be expressed in this cell line in any of the published studies that I had found. This tells me that there is a possibility that kisspeptin has some sort of effect on this cell type and gives me many ideas for where I would like to head next in terms of this project. I'm currently rerunning a repeat trial of this assay in order to bolster the data, and I'm also adding an additional condition where the cells are placed in an orbital shaker for various time points, and this is meant to simulate mechanical stress. This will tell me if mechanical stress in this specific bone cell line has any effect on GPR54 expression. Being that I've only tested this receptor expression in one cell line, I also plan on repeating the series of experiments using an osteocyte-like cell line, MLOY4, as well as a different osteoblast-like cell line that comes from more of a weight-bearing bone. The final goal I have for this project is to treat GPR54 expressing bone cells with kisspeptin and then investigate whether this would induce expression of proteins that are normally activated during bone cell differentiation. I can't wait to keep going with my project and continue learning about this fascinating new role that kisspeptin might play. So thank you for listening and taking the time on my presentation. Have a wonderful day.